that. Let's do that. Meet these people. Fantastico. Let's get to work. So, how's everybody's day going? Anybody?
Good, good, good. Hot, but good.
It's hot, right? Tell me, because you unwittingly fell into the trap. Okay. Have you had lunch today?
I have.
And that means you're not feeling all that hungry right now, correct?
In the heat, sometimes I'm always hungry.
Oh, so tell us your favorite food.
Wow. Salads? A good salad?
That sounds interesting.
Okay, chicken wings.
There you go. Fantastic, right? And the moment you say chicken wings, what do you feel inside of your mouth?
Saliva?
Fantastic. And how about inside your stomach?
Churning?
I thought you said you ate lunch. Like, see, salads aren't all that filling, are they?
I had yogurt with a chicken in mine.
Okay, there you go. Fantastic. So the point is this, right? There are some mechanics that are involved in, let me see this. Okay. There are mechanics that are involved in actually how we will acquire materials and bring them into our digestive system, right? But at the end of the day, what's really happening is moment to moment, your brain is scouring the known world and going, where is my next allotment of energy going to come from? And that's one way to think about your digestive system, is that if your brain is telling your body, it should be looking for and then accepting materials that can be converted into energy. And in this case, energy in our class, of course, is glucose, C6H12O6. And it wouldn't matter actually what you ate, Desiree, okay? Honestly, Mother Nature is going to take whatever you ingest and she's going to try to turn it into some intermediate that will lead us to the production of glucose. Now, the whole point of asking you what you ate initially is that's actually how the chapter finishes off. It's what if we tell you the mechanics that are in place for all of this to function, right? What happens when we actually start thinking about how it is all integrated? Okay, so one way to think about this, one way I would suggest that you think about the digestive system is to first take it apart before you try to integrate it. And pretty much I've just drawn the digestive system for you there, where the beginning of this line here is your mouth. It's got a set of teeth inside of it, right? And then it's going to materials put inside the alimentary canal, and then it's going to be sent to your stomach, where a bunch of stuff's going to happen to it there. True chemical digestion actually occurs inside your small intestine. True absorption of materials occur inside your small intestine. And then we use our friends, the bacteria in our large intestine, regulate water reabsorption and all of that before everything is just sort of like dumped off someplace behind a tree, right? No, nobody wanted to respond to that one. They're like, you're talking about pooping, that's bad. Well, you all have to do it, otherwise you'll die from impaction. So you don't want to die from impaction, so you have to poop. But here's the sort of the first nudge, nudge, hint, hint, wink, wink of chapter 24. And that is this. Because this information is so daunting, what I want you to do is read the text, but when you get lost, you're going to notice that there are tables to each section of the digestive system. So what happens inside the mouth and the throat? What happens inside the esophagus? What actually happens inside the stomach, small intestine, and large intestine? These are broken up into unique tables that have distilled all of the information into specific locations. And let me give you one example that I think you will find very, very helpful before I start the lecture. Inside the 15th edition, on this day, 7/23/2020. We find, where the hell is it? Sorry, today feels like Friday. I don't know about you guys, and I know it's only Thursday. Table 24.3. It's on page 919, and there are several before it, but I think it's a really good example. So if you have the textbook open in front of you, great. If you don't, what you're going to notice is what they do is they break down where and what cells are doing what work. And this is going to help you put the entire story together, because you're going to see that I use this pretty general.
I'm sorry, you said 2413?
24.3. It's on page 919.
Okay.
Okay. And this literally gives you, so let's imagine it's Sunday morning, you've taken the quiz, you're screaming at me and Valhalla all at the same time, right? And you're like, how am I going to get all of this information inside of my head? And this is literally where I wrote a great number of the true and false questions on the exam. So not just this one table, but the tables, okay? But at the end of the day, remember what you need to do is you take the pieces of information from the table and sort of you'll have to synthesize it all together at the end. Now, having told you that, I'd like to begin the slide presentation. Okay? And that's because there's two groups of organs that actually compose the digestive system, right? So first there's the gastrointestinal tract, that alimentary canal I was telling you about, the mouth, most of the pharynx, esophagus, small intestine, stomach, large intestine. But there are also the accessory structures that people tend to forget. And I don't have teeth, so I don't care if you remember about teeth. But there are people in this class who are studying to go off to become dental hygienists and stuff like that. So we have to spend some time talking about what is a tooth. Likewise, what is your tongue actually doing in all of this? And have you ever noticed that more often than not, when you're chewing your food, you do not bite your tongue? Except for that moment you got distracted when you were still chewing. Then something bad happened, right? Yes, no, maybe? Salivary glands. What is the purpose of those salivary glands? Likewise, the liver, it gets its own chapter. That's how important it is. But that's next week. The gallbladder, we cannot have a functioning digestive system without a gallbladder and a liver. Not to mention the enzymes that are made in the pancreas. So even though we actually think about what's going on primarily in that little cartoon that I drew, you'll notice inside that cartoon, I did not put any of these accessory structures. That doesn't mean don't study them. It means your textbook presents them to you and it's sort of like, oh, here's this piece and then there's this piece over here. Okay. Now, having told you that, cartoon of the digestive system, I happen to think my cartoon is much more informative than this one, but there's a lot of detail here. And this is the anterior view of a human that's been ripped open. This is what you'd see if you looked inside of yourself right now. Maybe not as much adipose tissue, or maybe not as much liver, depending on how much you've been drinking in your life so far. Those individuals who have been drinking quite a bit of alcohol since COVID came along, there's a whole series of papers out about how it is sitting around inside your house and drinking alcohol is not good for you, but you already knew that, right? Point being is the increased alcohol increases the size of your liver, but we'll come back to that later. The purpose of your digestive system, it's to ingest materials that need to be catabolized. That's what it's doing, right? How it's doing this is it's going to secrete a series of materials on whatever it is you've ingested. Chelsea, are you with us, seeing as you like teeth?
Yes.
Fantastic, Chelsea. What did you have for lunch today?
I had waffles.
Kick ass. With syrup or without syrup?
Without.
Did you have any like meat products bathed in butter or just sort of like waffle?
I usually eat it with butter, but I'm trying to eat healthier.
Oh, okay.
Yeah.
But we can work with this. Are you willing to work with this?
Yeah.
Fantastic. Did you use a knife and a fork, or did you pick up the waffle and just ingest it?
I picked up the waffle.
Awesome, right? And while you were picking up that waffle, I'm sure you noticed, just as Desiree had mentioned a moment ago, that the salivary glands released a series of materials into a space. And in this space, and in its case, the luminal space is the oral cavity, right? Yes, no, maybe?
Yes.
Okay, cool. She's like, yeah. And now you're thinking about the waffle, wishing you had some more, correct?
Yes.
And while you were chewing it, what you're doing, of course, is mixing all of these materials in with the actual, I don't know what a waffle is. Were these like pre-frozen Pop-Tart like waffles, or did you cook these yourself?
Yeah, the frozen ones.
Yeah, so there's probably very little nutritive value there, unless you like went to Trader Joe's and got the like oats and bark tree waffles, which taste horrible, right? But they're a little bit healthier for you. So which ones did you eat, Chelsea?
I ate the frozen one.
Yeah, but from like Lego or Eggo or whatever they are.
Yeah, Eggo.
There you go. Okay. Don't worry, you won't die a swift and painful death. But what's actually happening there, of course, is now, because you've mixed all these materials in, your teeth, you get that? Your teeth are beginning to mechanically break up the material and fold the liquid inside of it. And the enzyme, the major enzyme that's going to be released inside of your mouth, is going to be salivary amylase. And what happens is that salivary amylase, as I said, is going to catabolize things. But the major reaction that's now going to happen, of course, will be a hydrolysis reaction. And this happens each time we move into a different segment of the digestive system, until at number 5 here, we get to the absorption piece, where materials pass into your blood or your lymph system. Because you said you liked butter. Now you know why I'm asking you about butter, because that butter would have been transferred into the lymphatic system. Okay, as opposed to all of the other fake materials inside of your Legos or your Eggos or whatever they are, which are going to travel into your hepatic portal system. I didn't see anybody writing anything down there. Hepatic portal system. And eventually all of this gets removed in your body. Chelsea, thank you very much for playing along today. Are you crying yet?
No.
Good, thank you. Okay, cool. So how does this all work? Okay. The walls of the GI tract are pretty much made up of the same basic 4 layers. You'll have a mucosa, you'll have a submucosa. Inside that mucosa, obviously the epithelia is for protection, secretion, and absorption. And the lamina propria is the connective tissue that's going to hold it all together. But remember, we also have, look, reviewing for chapter 22, mucosal associated lymphatic tissues. So even though Chelsea ate what she thought was gamma irradiated, no nutrition, Eggos from Kellogg's, actually all the bacteria and stuff that were on top of it that were processed afterwards, okay, she ingested those. But because she had mucosal associated lymphatic tissues, those bacteria were identified as foe and destroyed. That makes sense? You're like, is all of my food covered in bacteria? The answer to that is yes. Okay, so that's why you have that mucosal associated lymphatic tissue. You also have this layer that's called the muscularis mucosa. And in some parts of the digestive system, you may need to change how the epithelia is actually moving. So by either increasing or decreasing the circumference of the lumen, you can now change what's actually happening inside that space. The submucosa is going to be made up of connective tissues. We'll have blood vessels, lymphatic vessels, and nervous system innervation, and that's what the submucosal plexus is. Whoops, sorry, wrong thing. And then below that is the muscularis. Nudge, nudge, hint, hint, wink, wink. There are some places where this is very important, but to propel materials through the digestive system, we utilize circular and longitudinal fibers such that it will now begin to push materials, starting from your mouth through the pharynx, the upper 2/3 of the sphincter, and all the way through to the anal sphincter. Okay. Now, when we're thinking about the serosa, this is the outermost covering, and this is actually how we suspend everything inside the peritoneum. Okay, and when we actually take a look at what this looks like, I think it's the next picture. So if here is moving from the inside out, here's the mucosa and that epithelia. Deep to that is that muscularis mucosa. Deep to that is the submucosa. And here are our circular, which are most deep, longitudinal are most superficial layers of musculature. And we have here those plexuses I was discussing with you. This suggests that our autonomic nervous system is responsible for regulating our digestive system, because you can't tell your digestive system to stop digesting food. So the enteric nervous system, I mean, I mentioned it way back in AMP1. You know, if you took AMP1 with me, you heard the story about going out to your favorite restaurant and getting sick and waking up in the morning and having to run to the bathroom and not being able to get your pajamas off in time and all this sort of stuff, right? And it's happened to everybody, but nobody will admit it, right? What happens, of course, is your enteric nervous system has an intrinsic set of nerves that are constantly assessing what's happening inside of your digestive system. And these two plexuses, the myenteric and the submucosal, these regulate how things move. And by myenteric, that's tract motility, whereas the submucosal plexus controls the literal secretion of digestive enzymes and mucosal secretions to keep things moving. Okay? Anybody ever worked with a patient who's been impacted before? No? Anybody even know what I'm talking about? They're like, you mean emotionally impacted? No, I mean, they're so dehydrated. They're so dehydrated that their body doesn't have anything to secrete using the submucosal plexus. So they literally, when you palpate their gastrointestinal tract, you can feel the bricks of tissue that are forming where the fecal material is no longer leaving the body. So one thing you actually need to make sure you're always aware of is the hydration, especially of elderly patients where they form rapidly. For this to work though, you can't just sort of, how do I say this gingerly on our first 15 minutes of class, right? You can't just apply an enema and hope everything's going to work out okay, because if the tissue is backed up and you apply fluids from the inside, what will happen, of course, is the movement of the material now will rip the tissue apart. So slow application of intravenous fluids would be a better way to approach this. But if it's life-threatening, a little bit of water goes a long way. How we actually regulate this? Primarily, digestion is regulated through your parasympathetic nervous system. Sympathetic nervous system inhibits digestion. Okay. Elizabeth, have you ever had a big meal and gone for a run?
Oh, I'm sure.
And how did that go for you?
It probably was terrible.
And
I probably vomited.
fantastic, right? Now, you're not expecting to be talking about vomiting here in the first 15 minutes of class. You're like, oh, I know where this is going. He's going to have us throw up, right? But the question is, why are you throwing up, right? By activating the sympathetic nervous system, you actually stop blood flow to the digestive system. And by stopping blood flow to the digestive system, your body doesn't want anything in there that could create problems for you. So your body goes, what's this doing here? And then let's just imagine you had, imagine somebody threw up this morning in class and they had chicken Alfredo, right? They had chicken Alfredo and they'd been drinking too much alcohol, right? So, so you can imagine the mixture. It was Myers brown rum or something like that, and the chicken Alfredo that came out of their gastrointestinal tract. So Mother Nature needs to regulate using the parasympathetic nervous system to allow digestion to occur, and the sympathetic inhibiting effects of the enteric nervous system literally stop the movement of material through the normal movement through the gastrointestinal tract. Here's a wiring diagram for you. Just remember this, that the submucosa regulates the fluid portion, the secretions. Myenteric plexus regulates muscular movement, and the two types of muscles that are moving usually are longitudinal and circular movements. And you said usually, which means there will be an exception. Okay. not so important, but we divide the ventral cavity and the abdominal space into the parietal peritoneum and the visceral peritoneum, and you actually 5 major folds. Take a look at it. Yay, yippee yippee. If you want a way to remember it, adipose tissue is stored inside the omentum of your body. And if you think about Bart Simpson, what's Bart Simpson's dad's name? I don't know his name. Does anybody know Bart Simpson's dad's name?
I think it's Homer.
Fantastic. Homer, he drinks too much beer, right? And he ends up with this large, what you're actually seeing is the greater omentum and lesser omentum are now filled with adipocytes, okay? So what Mother Nature does is she takes what is extra energy, in this case, the calories and the carbohydrates of the alcohol, and stores it there. What exactly is your mouth doing? Okay, it's a buccal cavity. You already know that. And remember from last night, it's more beneficial for you to breathe through your nose than it is to breathe through your mouth, right? Because remember, breathing through the nasal cavity increases the humidity, mixes the air, yada, 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 blah, blah, blah. Okay, but you actually have a series of, that's supposed to say soft palate, not sought palate. Sorry about that, okay? And what happens, of course, is the cavity is literally figuring out just the size of materials that can pass into the esophagus. And you're like, my stomach is, my eyes are always bigger than my stomach. And if you've been really hungry, like you grab a piece of pizza and you push way too much pizza inside your mouth, and then you're like, until your brain determines that the size of the bolus is small enough to now actually pass into the esophagus. That's what is usually happening. And believe it or not, people have studied the average number of times you should chew your food. Does anybody know what that number is? Guarantee nobody does it. No. 30. You're like, but there's no way in hell I'm going to chew my chicken wings 30 times. I take one good bite, it's gone, right? But the problem is you haven't then created all of the surface area for efficient digestion. Okay, now this doesn't mean tomorrow start chewing all of your food 30 times. I can see you eating your yogurt and your nuts going, right? Like, this is such a waste of energy. Obviously, it's those things that require much more surface area. The salivary glands will release saliva. There are 3 major pair of salivary glands: parotid, submandibular, and sublingual. We'll cover those in lab, right? Where you want to be able to, if I point to a needle on them on a model that you're going to stare at from 50 feet away, right? Because we all can't get close enough to one another, right? You need to be able to identify where they would be found. Say, right? Here are your teeth. My teeth look nothing like this at all. Okay. And here, of course, is your uvula. And your uvula actually has a responsibility. It's detecting when something hits it. And likewise, your tongue and the linguinal frenulum. The linguinal frenulum is that little flap of tissue underneath your tongue. It regulates which way your tongue should be moving. Okay. You do not want your tongue going backwards inside your throat. The reason, of course, is it will suffocate you. Okay. So we align a series of teeth here, and these teeth are designed to either grind, perforate, and or just simply chomp through so that you can actually increase the surface area of materials. And here are those glands, okay? Here's the parotid gland on the side of your skull. Sublingual gland is underneath your tongue, and your submandibular literally is beneath your mandible, which makes up your jaw. Most of that is water. 0.5% of the material inside your saliva is ions, dissolved gases, urea, uric acid. For the purposes of this class, lysozyme and salivary amylase are the 2 most important things. It's controlled by that parasympathetic nervous system. And if you think about it, every time I've asked you if you're hungry, what you're doing is you're activating your parasympathetic nervous system to then begin the entirety of the digestive processes. Once you actually think about food, right? The next sensation was your stomach was preparing should something actually descend from your mouth, okay? But then somebody says, run for your life, right? Sympathetic stimulation now decreases salivation. Your stomach's like, I don't really care what you want to eat for dinner today, okay? Cool. Tongue maneuvers food for chewing, shaping the mass, forces food back towards the back of your throat, so you can actually now grab onto it using the musculature of the esophagus itself. Your teeth, these are an accessory digestive organ, and there are three major regions, again for Chelsea, okay? Here's the enamel that covers the outside of the dentin of your tooth. And you actually have roots that travel into those gomphotic joints that are held together by pigments. These periodontal pigments are those same pigments you played with when you were a kid and you're hoping for the tooth fairy to arrive and give you some money underneath your pillow. And you felt that last pop. That's what actually gave. Once we actually push food back into the back of the throat, remember last night I said you needed to walk out of that class understanding nasopharynx, oropharynx, and laryngopharynx. The oropharynx is where digestion actually will begin. You push the bolus of food into the oropharynx, and it now moves down towards the laryngopharynx into the esophagus. And the esophagus itself, look at that, secretes mucus, transport food, but there's no enzyme production and there's no absorption of materials. This is just primarily a transfer conduit. We're going to break this musculature into thirds, where the upper third of the esophagus is 1/3 skeletal muscle. The middle third is skeletal muscle and smooth muscle. And by the time you actually get to the upper esophageal sphincter, okay, and the lower esophageal sphincter, what's going to happen is that regulated movement of material is to send small amounts of food into your stomach at a piece at a time. Here's what it looks like. Here's non-keratinized stratified squamous epithelia sitting on top of a lamina propria, and you have both the muscularis circular layer and the muscularis longitudinal layers here all being held in place with that adventitia. Notice no cartilage. Okay, remember in front of this, that's where you find the trachea. The esophagus is found behind it. So actual swallowing itself involves the three stages where it's voluntary, pharyngeal, and esophageal. That's the, okay, I need to get a drink of water, so you voluntarily try to pass the bolus of food to the back of your throat. Once you've moved into the pharynx and into the esophagus, that's pharyngeal and that is involuntary, and there's no way to control the esophageal movement once it passes into the esophagus. The mechanism that regulates this is what we call peristalsis, okay? Where literally the changing of the diameter and flattening of the tissue is what pushes the bolus forward. Now your stomach, okay? It has 4 different parts to it. We'll go over that inside lab. More importantly for you is it has a series of exocrine glands and endocrine glands. You're like, what? My stomach is an endocrine organ? Secondary, okay? Where it's going to make mucosa, okay? Or it's going to make a mucus to make sure that the tissue of the stomach is potentially protected. Parietal cells, which are going to make 2 molecules. One is intrinsic factor, and the other is hydrochloric acid. Yes, and you're reading that properly. Hydrochloric acid is what's being made here. And then you have a series of cells that will make pepsinogen and gastric lipase. So a whole bunch of stuff is about to happen here. And really the way it works is whatever you started digesting inside your stomach, inside your mouth, by the time it gets to your stomach, it will be inactivated. And then, because your parietal cells make hydrochloric acid, they then begin to change the shape of pepsinogen and gastric lipase, so these enzymes can start working. So this is an example of a pH-regulated mechanism for digestion to occur inside your stomach. It all happens because G cells are stretched, and these G cells release this molecule called gastrin. And inside your textbooks, I don't think I have a slide of it here. I don't. Do I? I do not. Inside your textbook, if you have the 15th edition, I want you to turn to a specific page. And that page is 919 inside your text. And they have this beautiful little figure, 24.15. 24.15, where they describe the regulation of hydrochloric acid. And inside this figure on the left-hand side, in yellow, you see the lumen of the stomach. And in blue on the right-hand side, this is the interstitial fluid, where notice what's being presented on the outside of the cell. Acetylcholine binds to the acetylcholine receptor, which means the parasympathetic nervous system has been activated. Gastrin is now actually bound to the gastrin receptor, which means there's something that's inside the stomach and stretching the stomach. And then finally, you actually have histamine, and histamine is being made from the cells in that submucosa to now think of this as 3 layers of correction or 3 layers of regulation to ensure that hydrochloric acid is only made when it's needed to be made. Now, Desiree, you can find out what your child wants. Oh, just don't hit them on the screen. That's all I ask. Good, cool. Okay, so now notice what I did. I said, oh, okay, look, there's a really sort of simple thing. Parietal cells make hydrochloric acid. But then I stressed that there is an endocrine function to this. And then what you see is the endocrine function works with the parasympathetic nervous system to regulate the production of hydrochloric acid. Okay? Now, what's going to happen here, of course, is the stomach's going to be made in a slightly different way. It actually has 3 layers of musculature. Okay? So you mix all this stuff together and sort of like, I don't know, a bread making machine, right? The blades spin around. Your stomach does pretty much the same thing. It uses the circular, longitudinal, and now inner oblique layer of musculature, and now folds all of this material together. It continues to do this until everything has been liquefied. Okay? Now, you should be thinking to yourself, I know that's not the case, because the last time I ate corn on the cob, what happened? There are some things that are not liquefied by hydrochloric acid, okay? Remember, we do not digest some forms of we don't digest cellulose, period, right? So what Mother Nature does is, in that instance, she uses those materials as an irritant for your small intestine, so that whatever you ate along with that corn comes out rapidly. Okay? That's the whole purpose of your salad, believe it or not. There's essentially no nutritional value at all. It's just a good irritant for your small intestine. Okay. Cool. So here's a cartoon of your stomach. Here's the fundus. Here's the greater curvature. Here's the lesser curvature here. So greater on the outside, lesser on the inside. And what you see is you have the internal oblique. You have now the circular layer and then the longitudinal layer of musculature outside. And remember I said your stomach can be distended. It can be distended because it folds in on top of itself to create these modifications that are called rugae. And these are folded tissues. And these folded tissues can then be stretched. Think about, I don't know, if you celebrate holidays with family and everybody brings over much more food that can ever be eaten, but you try to eat as much of it as you can. And next thing you know, you feel like you want to put your feet up and go to sleep or anything like that. No, never happens to you. Well,
All the time.
well, the idea, of course, is all that gastrin, all that intrinsic factor, all of that breakdown of material is signaling your body, well, you shouldn't get up and run right now. So you should just chill out for a while and let us liquefy everything you put inside your stomach. Okay? So how does your stomach actually do this? It actually has an array of cells. And so far, I've only pretty much focused on those parietal cells because they make that hydrochloric acid, and nudge, nudge, hint, hint, wink, wink. They also make intrinsic factor, but I haven't spoken about that yet because it's not working yet. Our chief cells, they're going to make that pepsinogen. And what do we remember about anything that's named gen? Fibrinogen, plasminogen.
Is it an enzyme?
It will be an enzyme once it's processed. So this is in active form, and then we're going to use that pH shift. We will process it, so we end up making pepsin. And pepsin begins amino acid or protein digestion, whereas the gastric lipase in the correct pH now begins to break down lipids. In one place inside your textbook, they say that there is a linguinal lipase that works in conjunction with your gastric lipase. They both require a pH shift to function, but there's only one sentence in there, so I wouldn't pay too much attention to it. Okay? And then way down here is that gastrin, that G cell that's actually making the hormone gastrin. So what's actually happening? So a way to think about this, of course, is the mechanical piece is the movement of the stomach with those three muscular layers, okay? And chemical digestion, not only by the enzymes, but also by the hydrochloric acid, which of course is going to kill all of those microbes that were on Chelsea's Eggos or Legos or whatever they are. They're called Eggos or Legos? Which is it? Anybody?
Eggos.
Eggos, thank you very much. Cool. Now, small amounts of nutrition can actually be absorbed inside your stomach. You know, aspirin passes readily through the gastric mucosa into your circulatory system. Likewise, drinking on an empty stomach is never a good idea because it passes directly into your circulatory system, right? That's why everybody tells you to eat a big meal before you go out with your friends, so you don't end up doing foolish things that, like getting your tied around a telephone pole. The police have to come undo you later. Never happened to me, never. I promise it didn't. It happened to my other teammates, but I was like mortified that they actually did that to that poor soul. Anyway, okay, cool. You didn't need to know that story. I just felt like sharing it. Okay, cool. So we're going to transfer now to, if I told you a moment ago that we've now liquefied all of the materials that were inside your stomach, and now it's going to travel into your small intestine. What has to happen here is we have to figure out a way to offset what?
Can you say that one more time?
Sure. If we've just liquefied everything inside of your stomach, we're going to move it into your small intestine, what do we actually have to offset?
Pass it to the duodenum.
Well, that's where it's going to. You're completely correct. But I'm asking, if we created all of that hydrochloric acid inside of your stomach, what have we actually made?
An acidic solution?
Yeah. Okay, and remember, your stomach is designed to withstand that acidic solution, but your small intestine is not. So what Mother Nature does is she makes a series of buffers inside the pancreas, and she makes a series of buffers inside of your liver. She stores them inside your gallbladder so that when materials actually enter in directly into your small intestine, materials from your pancreas and materials from your gallbladder will now mix with the acidic materials coming from your stomach. Mother Nature regulates the volume. It's about 2 tablespoons worth of materials that move into your small intestine at any given point in time, increasing
would
the
that
likelihood.
be basic material?
I'm sorry?
Would it be material that's basic?
Yes. Well, I mean, it's pretty much concentrated bicarbonate is really what it is. Okay? And if you remember way back, it seems like a lifetime ago. My God, it really does seem like a lifetime ago. Remember back in chapter 18 when we were looking at the pancreas? You're like, yeah, you're lying to me. You don't remember anything. But remember, we paid attention to the islets of Langerhans, and then I said you had all these acini cells. We would come back to those acini cells. Tonight's the night we come back to those acini cells. The acini cells are making not only digestive enzymes, but they're also making all of that bicarbonate. And that bicarbonate is now what's mixing with that fluid that's just come out of your stomach. Okay? So pancreatic juice, okay, it's mostly water, primarily sodium bicarbonate, and this is where we actually have the vast majority of enzymes that are released to mix with the contents of your stomach. And before you freak out when you start reading this chapter going, do I need to know all of these names and all of that? No, in fact, what I recommend you do is you try to look at them as though which ones are proteolytic, which ones break down proteins, which ones break down lipids, which ones break down nucleotides, as well as amylases. Okay, so carbohydrates, proteins, lipids, and nucleic acids. I will, in the exam and on the final, I believe we do this as well, we will say proteolytic digestion with trypsin, okay, would give us what products. Okay, so it's not a biochemistry class. You don't need to learn what each and every single one of these enzymes is doing. But I do want you to pay attention to the fact that they all come out as precursor enzymes. So they actually have to be mixed. The pH has to be appropriate. They're all then converted into their active form. And then they begin to break apart the different amino acid lengths, regardless of the type of protein you had that day, or regardless of the type of lipids you ingested that day. Because think about it. You had a salad today, right? And even though you're like, I got no nutrient value out of that. Well, you ripped apart plant leaves, and inside those plant leaves, there's still proteins. There's still nucleic acids. There are still carbohydrates. Those things that can be digested will be. The things that you can't digest, such as those cellulose monopolymers, those will just pass through your body, and maybe some bacteria will try to modify them, but on the whole, they won't. How is the liver and the gallbladder actually functioning? Well, the liver, who cares? It's the heaviest gland inside your body. Remember I said I'd return to that. And I know we don't have any, at least not during class time. I don't see anybody drinking bottles of straight gin while I'm lecturing, right? Maybe the ones who aren't showing their faces are doing it. But at the same time, what you actually see at an autopsy with somebody who would say you're an alcoholic is the cirrhosis of that tissue. What's actually happened is the inflammatory response to those tissues has damaged the connective tissue. And that's why you actually see the enlarged structure. It doesn't mean your liver is functioning better. Okay? The hepatocytes are the major functional cells of the liver itself, and there it's going to secrete bile. That bile is the digestive material that is mixed with a series of salts. And these are choline-based salts. They help with emulsification of lipids, and they also have bicarbonate mixed through them. So you have buffering components, and you have the bile breakdown products. And by that I mean we will need to increase the surface area of lipids using bile salts. Okay, so think about, so remember I asked Chelsea if she had butter on her waffles, right? Had she ingested a large amount of butter traveling through her digestive system, Mother Nature would have to make that more soluble by taking bile salts, bathing it through that butter material, and now what happens is it breaks it up into tinier and tinier droplets. And now those droplets your body can metabolize. All right, we don't need to worry about the hepatic sinus because there are 3 different ways of describing how these all work, but more importantly for you, like here's what you'd see inside a slide. More importantly for you is I've already told you something you didn't really pay attention to, and that was materials traveling out of the small intestine travel to the liver via the hepatic portal system. And every time we've discovered a portal system in this class so far, you've gotten lots of test questions on them, right? So here's another portal system for you. And what this means is Mother Nature takes deoxygenated blood from your small intestine, mixes it with oxygenated blood from your hepatic artery, and then she lets the hepatocytes actually do their job. And what they do is they collect the materials that are being made, okay, through these bile canaliculi, and subsets of materials be metabolized inside your cell. That's chapter 25, chapter 25. But what we need to think about is the materials that have been collected now into these bile canaliculi. So we come back to the liver inside chapter 25, but the important part is this. We've mixed deoxygenated blood with materials that have been digested in the small intestine, and we're mixing them with oxygenated blood inside the liver so the liver can do its job. Notice I haven't told you the mechanism yet, so we're not finished with the liver yet. So that gallbladder, okay, it actually has smooth muscle structures. Does anybody have any gallstones in the class? No? There's nobody who's going to out themselves. I was like, I'm suffering from gallstones. Of course, the first thing I'm going to tell you is drink more water, okay? Because gallstones, these are concentrated structures where what happens is you have calcium salts and those choline-based salts. They all just sort of form on top of one another, and then they just get too big to pass through the lumen of the tubules. Painful. If you don't take care of it, really painful. Here's that hepatic blood flow. Remember I told you we're going to mix oxygenated blood and deoxygenated blood traveling from the small intestine. Those pass to those liver sinusoids. We don't care about the rest of it because you already know it, right? Deoxygenated blood ultimately travels to the right atrium of the heart. Wham. What is bile? Okay. Hepatocytes will make roughly 800 to 1,000 milliliters of this for a healthy human being. It's mostly bile salts. There's that cholesterol we learned about inside chapter 18. Lecithin, bile pigments, and several ions. These are actually helping to metabolize lipids. Okay, the bile pigments you're just going to metabolize inside your lower digestive system. But the principal bile pigment, bilirubin, right? Remember that from chapter 19, okay? Will give us the breakdown product stercobilin, okay? Which ultimately, that's how we changed colors in that original cartoon back in chapter 19. That gets us to our small intestine, okay? So I've already discussed the small intestine with you. I told you that's where true digestion is going to occur. We're going to use those enzymes that were released from the pancreas, as well as the contents from the gallbladder. And in doing so, what we want to make sure happens is that the mucosa, which are going to be absorptive cells, and the cells that will release intestinal digestive enzymes that are going to sit on top of this, will now pass in a sequential manner through the duodenum to the jejunum to the ileum. So we go in a sequence, in order. Okay? Mother Nature increases the surface area of the small intestine by creating these structures that are called plica circulares. And what that means is, because they cut this tube in half, which you guys are going to do in lab, right? You're going to take your pig, you're going to cut through its entire digestive system. You're going to see all of these folds. You'll take your finger and you'll run it through it, right? And what you'll feel is what Mother Nature does is she puts microvilli on the surface of all of these folds, further increasing the surface area, because that's where the enzymes are actually doing their work. So as the liquid passes through, there's a likelihood that an enzyme that can catabolize whatever that material is will now break down the material and it will be absorbed. How does that actually happen? You can see inside this figure right here. So here are the microvilli on the absorptive cells. I'm pointing to right here. Deep to those are those goblet cells to make sure things keep moving along. And how you regulate the small intestine is through this population of cells that are called enteroendocrine cells. Dun, dun, bum, bum. Okay, enteroendocrine cells, these produce 3 important enzymes. They get their own table, folks. I'll even tell you the page so that you can say when you see them on your exam, you're like, he never told us about those 3 important hormones. On page, where is it? Come on. A 1,000 years later, on page 944, you can see the 3 most important enzymes in the digestive system. But you'll notice on our list, which says here, secretin, cholecystokinin, or GIP, on the list on page 944, it is gastrin, secretin, and cholecystokinin. So gastrin is going to work with secretin and cholecystokinin to regulate the amount of material that's moved into the small intestine. Now that's engineering. If you're not impressed with that, there's just, I don't know what to tell you. Find a different career. Okay? I'm joking about the find a different career. You can be a great practitioner in medicine and not appreciate any of this stuff. Okay? Woohoo! So here's the duodenum. Yay, pretty pictures of it. In lab, we probably will not be using microscopes because I don't know which one of you will contaminate somebody else. Instead, I will put together immunohistology slides for you to look at on PowerPoint and go, hey, I can tell this is a small intestine. Okay, it actually has a repeating pattern that suggests to you that is part of the duodenum, where these are the folds and the microvilli are sitting on top of those. So that intestinal juice, okay, it contains water, mucus, it's slightly alkaline to continue pushing off those hydrogen protons, but it's the brush border enzymes where at the absorptive cells, true digestion is going to occur, and you find a series of enzymes now. Notice their names are all impronounceable to anybody who doesn't really care about this stuff. I have a dare for people. Next time you get a pet, name it aminopeptidase. No? The pet fish named aminopeptidase. See how long that goes for. But the idea, of course, is regardless of the carbohydrate type, Mother Nature should be able to break it down. Likewise, peptides of 1 or 2 peptides in length are targets for these enzymes. And nucleosidases and phosphatases digest nucleotides and phosphate groups. How do you actually regulate all of this? We're coming to the end of me talking for the day, right? Okay, how do you actually regulate all of this? You regulate all of it because that myenteric plexus can break materials up into segments. It exited your stomach as a liquid. Now it's being broken down. Materials are being absorbed. Those materials that haven't been digested or can't be processed remain in the lumen of your small intestine. And this begins to form a semi-solid bolus again. And that semi-solid bolus is now going to move through the migratory motility complex, or your migrating motility complex, which is a type of peristalsis. And it's going to begin to push food toward ultimately the large intestine. Now, inside of all of this, the chemical digestion is being broken down. And here what I've done in this slide, you can go back and read it when you feel like it. If I use the word carboxypeptidase inside of a test question, I will tell you it is an enzyme that breaks down proteins, right? Ideally, though, what you see here is the pattern. We've digested nucleic acids, we digested lipids, we digested proteins, and we digested carbohydrates. Why would I stress that pattern to you? Does anybody remember what we built in chapter 2? We built carbohydrates, we built proteins, we built nucleic acids, and we built lipids. So the significance here is, as a living organism, we have the ability to build these inside of our body because we've ingested and then broken down these from other organisms or other inert materials, right? So that we can then use them ourselves to build the other materials inside of our body again. So not only is your digestive system responsible for acquiring materials to make ATP, but it's also to repair and replace the tissues that are functioning inside your body moment to moment over the course of your life. That's a big, warm, fuzzy, natural, Mutual of Omaha Wild Kingdom moment there for you, right? Most people think your digestive system is just to get energy. Remember, you need to replace all of the tissues that are dying in your body moment to moment. Now, how do we actually absorb all of this stuff? We're almost exactly where I wanted to be at exactly this time. I don't know how that happens, I swear. Do you guys ever lose track of time during lectures, or you're just like, would you just please shut up? No? Exactly. Yeah, no, you just want to say, just shut up. Anyway, but the idea, of course, is Mother Nature needs to not only absorb this material, but she has to absorb it in a certain state. It has to be processed to a place where it's actually usable in your body, right? If you've ever done any, does anybody do any large data science work? No. How about when your kids come inside of a room and they're all screaming at once? What do you actually have to do then?
Scream louder.
Okay, to get them all to what? Keep quiet, and then you tell them what? One at a time, right? You first, you next, that. So you can process the information. What happens is your body does exactly the same thing with monosaccharides, amino acids, separating out individual amino acids from dipeptides and tripeptides. Let me show you that. This is a figure inside your textbook. I thought I blew it up, but I didn't, okay? What you can see here is how this actually works. This works because the intestinal cells are designed to transport only a subset of the material that's out here. Once you've actually gotten a single amino acid, it gets absorbed immediately by diffusion. Once you actually have glucose or galactose or even fructose, it can be brought into those cells, but notice the mechanism. So glucose and galactose could be secondary active transport with sodium. Therefore, we maintain sodium inside of our body. This is figure 24.2 if you're lost. Okay, cool. Okay, good. Yeah, cool. So the purple structures, right? It's glucose and galactose and fructose. They have slightly different mechanisms, right? Likewise, when we go down here to amino acids, dipeptides, and tripeptides, they have slightly different mechanisms as well. Things get really weird when we finally get to the lipids. Nucleic acids are absorbed immediately, okay? We form these structures that are called micelles and monoglycerides, which can then be transported into the epithelial cells. Once inside the epithelial cell, way back when inside of AMP2, when we built those lipids, remember we built triglycerides with a glycerol backbone and 3 fatty acid tails? Well, we told you those are transported. We didn't tell you how and why. What I'm showing you tonight is for us to transport a triglyceride, we have to create this structure that's called a chylomicron. Like, what? Remember, these are fatty acid structures. They're not soluble inside the plasma of your blood. So you put them inside a chylomicron, you transport that chylomicron into your lymphatic system. And your lymphatic system now has the responsibility of diluting that, okay? Such that by the time it finally gets back to your venous blood return, it's been diluted, and now your liver can begin to break down all of these structures. A way to think about it is this, and I've seen it firsthand, so believe me, you'd be completely grossed out if you saw it yourself. Go out, get 2 pieces of pizza, eat them, wait about 30 minutes, get your blood drawn out of your body, take 50 mils up. Tell me what color you think it is. Anybody, be brave. Again, so the scenario is this. Go out, eat 2 pieces of pizza, okay? Wait about 30 minutes. Go get your blood drawn. 50 mils, it doesn't have to be 5 mils, will be plenty convincing for you. I want to know what color you think your blood is going to be.
Is it milky white?
It is milky white. Kick ass. Desiree's like, I'm so on top of this. Why is it milky white?
I don't remember why. I just remember it being milky.
It's milky white because all of those chylomicrons, all of that surface area from all of that fat now has to be passed through your entire circulatory system, and then it's slowly metabolized by your liver. Okay? I had a friend who was he was doing research inside a virus lab down the hall from me, and he every Friday he would look for blood donors, and in the background of this story is the same person's dying of adult myelogenous leukemia, right? So that's the background story. And he asked for a blood donor, and it was after our normal Friday afternoon meeting where they fed us pizza. And he's like, "Hey man, I need some white blood cells." And I said, "Sure, no problem, Steve." And we went back after the meeting, and he tapped my arm, right? And so we had somebody come in, took, I don't know, 100 mils of blood out. But when you're talking with somebody who's dying of leukemia, what color is their blood? It's white, right? So just the collective feeling inside the room was like, no shit, you're fucking dying too? And I was like, no, I'm not dying too. At least I better not be. I was like, all I wanted to do is give you some blood so you could finish this goddamn paper before you died. But, and he did. He did finish the paper, and yes, he did die. And I still miss him to this day. But the idea is that Mother Nature needs to create that surface area so she can metabolize all of that energy slowly. Okay? With me? Cool. Woohoo! Yeah, nobody else wanted to hear that story tonight. Sorry. It's just my way of remembering them each year.
No, that was interesting. I thought that was a quick trick question.
No, not at all. Not at all. Yeah.
Super cool.
Yeah. So you're also absorbing other materials here. Electrolytes, vitamins, water is being mostly absorbed inside your small intestine. The electrolytes, notice the major ion that's being reclaimed here is sodium. Remember, for you to maintain your structure as a bag of salt water, you need to maintain that sodium inside of your body. Likewise, fat-soluble vitamins, A, D, E, and K, are taken up with lipids inside my cells, inside of your body at this point in time. So this is where I wanted to pause for the evening. So I'll pick up here tomorrow night, and what we'll do is we will finish this chapter tomorrow, and I will have put together a review package for you guys that will be based on the bullet points from the final exam, and I will intersperse the materials that I know you should be focusing on for your third exam inside Bio 212 at Norwalk Community College, where you're having all of the fun of your life right now. Okay, so now, did anybody notice that your take-home quiz is up there, number 1? And I'm going to unblind, I think it unblinded at 6 o'clock, your online quiz as well. Remember, the online quiz is due Sunday morning. You get 2 stabs at it. Go get an A. Go get an A on your take-home quiz. And I'm going to stick around for as long as you guys need to answer any questions you have right now. Any questions? No? Like, I want to get to dinner. Chelsea, you still want Eggos?
No.
Did I ruin your Eggos on you? I'm so sorry if I did.
No, not really.
Okay, you telling the truth?
Yes.
Okay. Well, I hope you guys have a wonderful night. I will see you tomorrow. If you have questions, just say, hey, you got a minute? And I'll stick around. Okay, cool.
Thank you.
My pleasure. Have a wonderful evening.
Thank you.
Thanks.
Au revoir. Au revoir.
How do I get out?
It says end meeting on the lower right-hand side. You've only been doing this for 14 days now.
Big
It's
red
all-
button.
There you go. Kick ass. Janelle, you still there?
I am.
Do you have questions?
No, not really. I'm just so lost, but I guess I have to find my head. I don't know. I feel like there's something in this class I'm not utilizing, so I don't know.
So there's no one resource that's going to help you be more successful, right? So
Mhm.
help me understand how you usually study for a class, and maybe I can offer some advice there.
Usually what I do is at first I will go into the chapter review in the back,
Cool. Okay, good. Yep.
and then I'll start reading.
Okay.
Reading to get more info and everything, and then start writing notes and taking notes. And then by the end, I'm hoping that I'll finish with my reading, go over my note, and I'm finding that although I did take the note, by the time that I'm done with all 3 chapters, I don't even have time to go back to the note. So what was the point of me taking the note to begin with? And I'm trying to do the test that's at the end of each chapter. Although I get them, I'm trying to go over them. Sometimes I get stuck. I get myself so involved into the question that's in the review test. It's just like I'm so all over the place. It's not even funny.
Okay. So why don't we try this? Okay. So have you started reading chapter 24 yet?
Oh yeah.
Okay, good. So here's what I recommend you do. I love the fact that you started with the review at the end, because that gives you the sort of broad overview. And now,
Yeah.
imagine, if you will, I only spoke about specific things this evening, right?
Yes.
So if you read only those pieces of the text,
Mhm.
okay, because you only have a certain amount of time, that's going to help you now write those notes for those specific pieces.
Okay.
Okay. And then what you have to do is you have to think like, so you have a general idea how the digestive system works. And then you have to think, okay, he spoke primarily about the regulation of movement through the system. And then he spoke primarily about the hormones that were being used,
Okay.
and then what materials were absorbed. And if you can teach those 3 pieces of information back to me, you're going to do a lot better. I think sometimes when people go to like the review questions, they do exactly like you do. They get lost in trying to figure out the answer, and that takes away time from making sure you can actually think through the material itself.
Okay.
Because so leave the questions that I give to you, the take-home quiz and the online quiz. Use those as your guide. So give yourself enough time so you can get that stuff done, and you're like, okay, I don't understand question number 4 on the take-home quiz. Turn that into a question and then send that to me, and then I'll help guide you from there.
Okay.
If you're taking on so much work that it's impossible to get all of it done,
Yes.
think about what I'm actually trying to do. I'm trying to give you the quizzes that are going to focus your attention on the materials that I know are most important to learn.
Okay.
Okay, because once you have a general understanding, it doesn't matter if you remember where mucosal cells begin and end in the small intestine. That's not important. Remembering what cholecystokinin and secretin do, that is important. Okay?
Yeah. Like last night, I spent so much time. Like I said, I'm going to email you the take-home quiz, which I already did it all last night.
Awesome.
But now, oh yeah, I mean, I have the time. It's just like I'm finding myself. Where do I pick up? I mean, how do I get my brain to process everything? It's like, okay.
Do you ever go for walks?
Oh yeah, I do. Mm-hmm.
Fantastic. On your walk, imagine you and I are having a conversation, and you're teaching me what you know. And when you stop, and you're not making stuff up, right? You're like, okay, I know I can get comfortably to here. That's when you go back and you open up the textbook again, and that's where you start. And that's how you keep yourself organized.
Okay.
Okay, so when we're thinking about, say, the immune system, we have an innate part and we have a specific immune system. Teach it to me like that. Teach it to me as the non-specific responses are skin, mucosa, macrophage.
Okay.
Keep it really simple, and that's going to help you remember it. Okay?
Okay.
And when you get overwhelmed, Don't be afraid to just get up and walk away from the table. Go for a walk around the block and come back and start again.
Yeah.
That walk is more important than you think it is.
I know, like to clear